Influenza is a viral infection of the nose, throat, and lungs that causes the symptoms of fever, chills, runny nose, sore throat, cough, muscle aches, and fatigue. The influenza virus is extremely small in size and can be viewed only with the aid of an electron microscope. The genetic material inside the virus contains the information from which it makes multiple copies of itself. The genetic material is enclosed in a hard protein shell which provides protection as the virus moves between the humans or animals that it infects. An outer envelope is also present which allows the virus to infect other cells by merging with the outer membrane of these cells. Certain protein molecules known as spikes project out of the outer envelope and could be of two types. H spikes, which act like keys, allowing the virus to enter the cell, or N spikes, which allow the newly created viral copies to break away from the cell to infect other cells. Flu viruses could be classified based on the type of H and N spikes on the surface. For example, H1N1, H5N1. The virus is spread through droplets from an infected person or animal which contain the virus and linger in the air for a while, or get the deposit on the surfaces, thereby contaminating them. When an infected person sneezes, coughs, or drools saliva, droplets carrying the influenza virus are formed, which eventually find their way to the mouth, nose, and lungs. Once inside the body, the virus comes in contact with the cells in the nose, throat, and lungs, with the H spikes allowing the virus entry into the cell by inserting into the receptor molecules on the cell membranes, much like a key in a lock. Once inside the cell, the virus travels to the nucleus by the means of a protective sac created from the membrane of the cell that it infects. The viral envelope and the cell membrane sac that unite, allowing the viral genetic material to leave the sac and enter into the nucleus. Inside the nucleus, the viral genetic material hijacks the materials and energy that is readily available to make multiple copies of itself. Some of this genetic material moves out of the nucleus and attaches to the ribosomes in the cell, providing them with the necessary information to manufacture viral proteins such as the H and N spikes. The Golgi apparatus that carries these H and N spikes as vesicles, which will merge with the membrane of the cell. All the necessary elements to put together a new virus gather just below the cell membrane, eventually resulting in new created virus budding off from the cell membrane. The virus, however, remains connected to the cell membrane through the attachment of one of its H spikes to a receptor of the cell membrane. The newly created virus is eventually freed from the cell when one of the N spikes cuts away the attachment to the receptor, thereby freeing the virus to infect other cells, ultimately making the person susceptible to the flu. Some of the commonly used drugs for the treatment of flu include oseltamivir, which is taken orally, and zanamivir, which is taken through inhalation route. Both drugs act by blocking the action of the N spike, thereby preventing the newly created viruses from detaching from the cell. The best protection against flu is to get flu vaccine, which is available in two forms: injectable, containing dead strains of virus, and a nasal spray containing live but attenuated strains of virus. Administration of flu vaccine results in an immune response being mounted with antibodies specific to flu strains that the body was exposed to, developing within 2 weeks. These antibodies get attached to each flu virus, thereby preventing their subsequent attachment to the body cells. The antibodies are also capable of attaching to more than one flu virus, causing them to clump together to be eventually engulfed by macrophages. A future infection by any of these strains of flu virus would result in body's immune system immediately identifying and destroying the same, thereby providing the necessary protection to the body. Administration of flu vaccine is advised every year for continued protection against the newer strains of flu virus.